There's only one way to do it. You need to let it grow. You need to let an amount of information unfold that you then need, if you wanted to describe that endpoint bit by bit, would be quite a lot. But if you would only want to describe the information needed to grow it, would be very little. But you can't predict from the little information what the endpoint would look like without actually growing it. And the genome is always just there. It's like a book that's always there, and you always just need to decide what to read in that book. And to access that book is just enormously complicated. You can't just open page 255. You literally need a very strange combination of, say, 30 different proteins that are super unlikely to ever exist at the same time in the cell. But if they do,
this is Brain Inspired.
That was the voice of Peter Robin Hiesinger, who recently authored the book The Self-Assembling Brain, How Neural Networks Grow Smarter. Hi everyone, I'm Paul, and today I talk with Robin about a handful of topics in the book. Robin is a neurobiologist, or a neurogeneticist more specifically, at Free University of Berlin, studying, among other things, how DNA and the developmental process encodes the wiring of brains in the fruit fly, Drosophila. The central theme of his book is that current artificial intelligence and perhaps current neuroscience theories are leaving out an essential part of what makes us intelligent, and that's the growth and development of our brain's neural networks, and in the case of deep learning, an artificial neural network. Robin suggests we don't yet appreciate how information, which begins at a relatively low level encoded in our DNA, unfolds and increases with time and energy as our networks are formed, something Robin calls algorithmic growth. And his claim is that it's essential to include a growth and an evolutionary selection process to get anywhere close to building something with human-like intelligence, if that's even what we want. So we talk about that central theme and many of the issues that arise out of it. And as you'll hear, the book is also a history lesson about the parallel yet independent paths of AI and developmental neurobiology. Show notes are at braininspired.co/podcast/124. Thanks to all my Patreon supporters. If you like this sort of thing and you want access to all the full episodes and to join our Brain Inspired Discord group, you can do that on the website also at braininspired.co. All right, enjoy Robin. Robin, I enjoyed the book immensely. So we're going to talk all about it. One of the things that I was surprised about when I read it, given that I'd seen one of your lectures, was the amount. So of course you dive into the history of AI because the book is all about how AI might be missing something, right? But what I was surprised about and thoroughly enjoyed as well was the history of developmental neurobiology and how you weaved that history into the history of AI, and how they happened in parallel, and of course, hadn't really spoken to each other for many years. So, and you did it in a format that was sort of a storytelling format, so it was very easy to digest. So, congratulations, and thanks for writing the book.
Oh, thank you so much for having so much good things to say about it. Yeah, it is a unusual angle, and it's because I'm coming from kind of left field, if you will, right? I'm a neurobiologist. I dabbled a bit in informatics and I dabbled a bit in philosophy as a student, but I'm running a research lab. I'm teaching undergraduate and graduate students, and we're publishing papers on how genes encode the information to wire a brain. And so the origin of this somewhat unusual approach and of the parallel storytelling of a field of neurobiology and artificial intelligence really kind of originates with me being, I guess, somewhat unhappy with my own field in biology, because, we started many years ago in the very successful molecular revolution to publish more and more papers on, individual genes, their products, and their roles in how the brain develops and how it functions. And it's led to a data explosion. And, a very good friend and colleague, Bassem Hassam, always says, we're we're increasing the amount of knowledge at an unbelievable pace, but our increase of understanding is not keeping up. And so I was wondering whether we're missing something. I was wondering whether we need some kind of theory, whether we need some kind of information theoretical background. What does it even mean that the genes encode the brain? I mean, that's obviously a very loaded, strange question. And then, of course, you can't avoid, in this day and age, being bombarded by the news about some kind of allegedly super intelligent systems, seemingly overtaking us next week. So what do they know that I didn't? And so that's when I started to feel like, okay, maybe these people know something that I don't. And I realized, and that's where the historical part comes in, that the history of AI is unbelievably fascinating. My goodness, these people have been at each other from very different fields for so many decades. And for most of those decades, they really wanted to have nothing to do with the brain or neurobiology at all. And all the successful approaches that we hear about today, this like unbelievable, success of AI that we have right now, is actually all based on what is called artificial neural networks. And they're closer to the brain than any approach in AI has ever been in history. So that got to be interesting, right? I mean, if they suddenly take an idea that we have been studying as biologists for so many years, and they can do with it things that we can't do, and then we feel we can also do some things that they can't do, maybe we should talk to each other. So this is how it started.
So I was imagining that as a neurobiologist, you saw the deep learning revolution, the neural networks, and essentially you said, whoa, they have a lot to learn from me. But in fact, it was kind of the other way around when you first poked your nose into it, thinking, what do they know that I don't know?
Absolutely. I mean, the idea that, they should be listening to us is dangerously close to ignorance in any kind of approach, right? Because you need to first understand what the other field is doing. And it's a very rich field. And so this is exactly why I had to go into the history. I mean, when I started, I just wanted to know, okay, what are they doing right now? What is interesting about this? And I found deep learning is really cool. The recent approaches in reinforcement learning make perfect sense to me. It even has an evolutionary angle. This is even more biological than anything, right? I mean, babies clearly learn with some kind of reinforcement learning, so that makes some sense. So that was all very good. But then you start to wonder, why are they doing certain things not at all that, I'm actually spending my life studying? And the thing that came to the forefront at that moment in my thinking, of course, was, wait, I'm a neurogeneticist. I'm studying how genes encode a growth process to make a neural network. And that thing's actually pretty smart before you start learning anything. I mean, we, can talk about examples later on from the insect world too. When a baby is born, it's not born with a random network that starts to learn. That's actually exactly what pretty much all deep learning approaches do today. There's no genome. There's no growth process. They're turned on to learn. And that made me then wonder, okay, why is it that they're doing one thing that we know from biology but not the other? Why is it that they initially didn't even want to use neural networks to start with? And when I started to understand their history, I had so many moments of, these kinds of problems I've seen before. We had that in biology. We had a time when people felt like the human brain is so amazing, and our thinking and learning, it got to, the genome, at the beginning it wasn't, of course, even not even known what the genome looks like. It had to be learned, right? And so this debate on learning, on nature versus nurture, on how much can actually get into a network via a growth process and to what extent you would even need it, has been there in the neurosciences as well, some time ago, but in one form or other, it's still there. And so this is very interesting, right? So to see then the parallels between those fields, to see how, from my perspective, to some extent, the AI community is actually retracing some steps that historically neuroscientists have taken, made me wonder that, there's something, again, that we need to talk about. We should. And I wondered how much we are, and it turns out there's very little crosstalk. And so this is how the book originated.
So I want to talk for a moment more about the book itself before we get into the ideas that are in the book, because I'm curious, for one thing, that was a very rich and detailed history that you told of the developmental process. Was that a lot of work, or was that easy to piece together? Because I know the AI history, or I knew the AI history quite well because it's been told a few times, but and I even learned more about it through your writing. But what I didn't know about was that rich history in developmental neurobiology. Did you just know all that, or was that a lot of research that you had to do to write that?
It's both. I mean, it came first, of course, right? I mean, it is my field, but you're actually touching quite an exposed nerve. I mean, if we go if you open textbooks, there's a lot of knowledge that is being communicated without communicating the field's history. Mm-hmm. And there are some debates that just appear ludicrous today, right? I mean, the whole debate, this is how the whole problem, how many of my lectures and the book start, is that people couldn't even agree whether a neuron is a physiological unit, because nobody could understand how you could have so many of those things wiring together, right? And so this seems, of course, it's a completely settled debate. Of course, we know that the brain is made up of neurons and they're physiological units, and somehow they need to connect up to make something that works and appears smart to us. But this you can only understand if you actually have the historical context. And of course, I knew some historical context, but probably not more than any other somewhat trained developmental biologist. And when I started worrying about my own field, this actually happened before any deep diving into AI history. And, where are we at right now? Why are we just collecting gene after gene and molecular function after molecular function and publishing more papers every year than ever in the history of developmental neurobiology before? And are we actually getting closer to understanding how that thing is put together? So when I started worrying about this and wondering about this, that's of course when I initially went back in the history of where this came from. And there's a very cool story to be told. That's of course in the book of an interesting breakpoint in history where a very famous neuroscientist, Roger Sperry, went ahead and said, you know what? The embryonic development will make a brain out of individual neurons, live with it. And there got to be molecular interactions that ultimately define the development and the growth of that network. And that was at a time in the 30s, 40s, and 50s predominantly, when most scientists, including the famous, even more famous supervisors of this gentleman, were on the other side of the spectrum and said, it got to be all somehow induced by some kind of plastic form of learning. And, it was a field of psychology. Brain wiring was at that time the subject of psychology and not the subject of, today, like molecular geneticists. They didn't even exist really at that time. And so this is where it all started. And it's been good and bad. Of course, we know that, there are a lot, some say an infinite number of instances of individual molecular functions in different contexts, in specific cells, in specific animals, at specific developmental stages. I mean, there's no limit to the depth of this that you can study and publish papers on. But how the genome actually does that? Famously, the genome contains 1 gigabyte of data. And there are people in the AI field, like Schmidt-Huber and others, who actually use that as an argument in saying, look, 1 gigabyte of data, that can't encode anything. So, we don't even need to look there. Clearly, we only need learning. We don't need the genome. But then again, of course, what we're all doing, maybe not looking at the whole forest at a time, but always like looking at some individual leaf or needle of just one tree inside that forest, is looking at how the thing unfolds. And we do know that an apple seed will grow an apple tree. This is the job of developmental biology. And the development of the brain is no different. And so this is basically where we're coming from. And this is where the history became so important and has a huge impact on what we do today.
So I will read from a quote from your book, I guess, to start us off here, because just dovetailing off of what you were just mentioning. This then is the core question of our workshop. What difference does it make for the intelligence of a neural network whether connectivity is grown in an algorithmic process that is programmed by evolution versus only learned based on a designed network with initially random connectivity? So you were just talking about people like Schmidhuber, and not to single him out because it's the entire AI community essentially, that it's after reading your book, it's just a curiosity that we all, I'll include myself, right? Because, at first pass, it's like you have this brain, you have the network, and then all you need to do is learn from there. And that's what intelligence is about. But your book makes the argument or asks the question that maybe the growth process itself from the genetic code is an important part of that process. And of course, there are debates in the deep learning field how important inductive biases are and the architectures that you use and whether that matters. But I would guess that you'd say that that's not enough.
Yeah, exactly. That's exactly what I would say. But I would also say it's a step in the right direction. Ideas that like convolutional neural networks that are basically mimicking a little bit of the wiring of a visual cortex in mammals, or the new proposal that just made somewhat of a splash by Jeff Hawkins in his Thousands Brains, is of course that, we got to design these things like the cortex. And then you have like all these cortical columns and then they can compete and vote and all these things, right? And these are all very good ideas. But they're basically coming like tiny steps back from like the purely random network. And of course, they're still designed. So in biology, how you got to the cortical columns in the first place was, of course, through a growth process. And the fact that there are cortical columns contains information. It's not just, a randomly connected network that we have in our brain. Otherwise, most of my neuroscience colleagues would be out of a job very quickly. They're studying circuitry. We're studying how the neurons are exactly put together. And it's fabulous. I mean, you can study things like motion detection. So it's a beautiful example by understanding how exactly different types of neurons with certain delays and certain, conduction velocities and synaptic strengths and then some state dependencies where you have some neuromodulators that whole populations of neurons suddenly have a lower threshold, all kinds of stuff. And when you see how all of this is put together, suddenly you understand you can even build a computational model based on that. And it tells you, this network can see motion. But this is very different from teaching a completely random connected network motion detection, right? So to teach the randomly connected network will not lead you to all the both cellular connectivity that is very specific that I just try to like in outlines describe, or the molecular aspects, which are things like I even mentioned something like a neuromodulator. These are molecules that are diffusing in whole areas of the brain and changing synaptic weights. And this is simply not included in the modeling of synaptic weight changes in an artificial neural network. So evolution has found solutions to problems that the brain can solve, like motion detection, like, you name anything up to cognitive abilities in the human brain or down in insects. My favorite example, as you probably have seen, is the monarch butterfly that flies these 3,000 miles. Right?
Yeah, you're crazy about butterflies.
Yeah, I really love those. They do so much. They're like half a gram and they can do so many things. If you wanted to train a neural network that is randomly connected to do all the things a butterfly can do to achieve butterfly intelligence, I can tell you we're far away from that. But without strain, this is the idea, right? So clearly, learning from biology has become more accepted than ever in the history of AI before. Jeff Hawkins famously was unhappy early on as a young person before becoming a billionaire that he wanted to learn from the brain. And, there's a whole history. And this is why I tell that history, right? In the book, in AI research saying, we don't need all this messy, wet stuff. And, it's all these idiosyncratic solutions that evolution may have found in the brain. We can design something from scratch that's better. And now we're at a time where we're kind of, dipping in. I mean, when I say we actually talk about the AI community, dipping into ideas from the brain, like, maybe we need cortical columns. But it's really just the tip of an iceberg, because the brain is not just simply cortical columns. It is all the molecular beauty that defines how individual neurons communicate with each other. And there is so much information, not just in the connectivity of a specifically wired network, but also in the molecular composition, that you cannot, if you want to simulate the way the human brain works or a butterfly brain works, just reduce to synaptic weight changes. And so this is where I'm coming from.
Well, so I don't know how much I agree with you that it's more acceptable now to include biological detail, because there's, and you document this in the book as well, there is a constant drive in the AI community to abstract out as much as possible, right? So the idea that you would need to include growth, and we'll talk more about the details there in a moment, the idea that you would need to include growth from the gene must be horrendously unattractive to someone in the AI community who's just trying to get their neural network to learn something, right?
Yes, it's very unattractive. Actually, we did the experiment together with colleagues, just how unattractive it is. If you just want to learn a specific task. And this brings us to a very interesting topic, right? I mean, artificial neural networks, based on reinforcement learning, have been at this point more successful in almost any individual task a human could do that I could imagine. I mean, obviously they can play better chess, they can do better visual image recognition, they can do, better solutions to the cocktail party problem, and auditory, whatever, right? So individual things, you can train these things to become better. If you now want to train the same network something else, you quickly have the problem of catastrophic forgetting. And the AI community is trying to address that with deep learning approaches, and there are some good proposals. Now, the bottleneck in teaching a network any of these things is still the training, obviously, because the design is largely random, right? I mean, you may have some pre-connectivity like in a convolutional or recurrent neural network, but really the key bottleneck is learning. So reinforcement learning takes a lot of time and energy to get that thing to be really good. Once it has learned it, you can just, deploy it. And then it can decide whether it still should learn anything or not. And, but once it has learned something, it just can do, right? It can recognize images. It can make predictions about, who should be your spouse or your next soap. Now, if you want to do this same job for a specific task, with a neural network that is not trained by learning, but that is trained by a genome that produces the network. And then once you grow that network, you have it, evolutionary selection for one that works, back to the genome and iterations of this process. Then you will soon find out that the time and energy that takes is even enormously bigger. So maybe it's really worth saying one more word about this. Right? Mm-hmm. The AI that we know today, everything in any of these big Silicon Valley companies that we're all familiar with, all the stuff that we're scared of or impressed by, it's all trained networks that have been trained in one or the other form, big data or reinforcement learning, deep learning stuff, right? But there is an AI community that actually does a very different type of learning of artificial neural networks. And it works like this. You take a genome. The genome defines, in the easiest case, directly synaptic weights. Mm-hmm. It's not how biology works, but that's the easiest, approach you can take. You can say I have one gene per synapse, if you will, right? And then you can basically fill the synaptic weights of a matrix of a recurrent neural network. And then you can have that thing do something. For example, let a little agent find a path through a maze or do image recognition, anything you want. And then if it did it well, then you take more of the genome that was at the base of this. And if it didn't do it well, you just mutate more of the genome at the base of this. Mm-hmm. And then you do it again. And then you do it again. And then you do it again. Just like backpropagation is an iterative process where you train and train and train in classic deep learning. And reinforcement learning is actually very similar to the process I just told you, right? Because you only learn from the end state of the system. So can you actually train a network not by learning, but by keeping on randomly mutating that genome that feeds the synaptic weight matrix? But that's still without a developmental process. And if you now add a developmental process, this thing gets very quickly, even for the biggest computers on Earth, out of hand. You'd have a genome that say, few hundred genes has not even been done. I mean, maybe a few dozens. And then you basically feed that into some gene regulatory network that may have to go through a few hundred iterations of a developmental process that leads to the numbers that you fill in the weight matrix for the recurrent neural network. Then you finally have a network that can, do a task, perform something, see whether it's good. And if it's good, it's good. You keep more of the genome. And if it was bad, you mutate more of it. Imagine this iterative process. So it's a huge computational effort. It's orders of magnitude more computational power needed to even simulate a laughingly simple version of a genome and developmental process in an iterative evolutionary selection process. And the outcome is, at this point in time, never better than the deep learning stuff. So therefore, nobody's doing it.
Right.
Well, that's not quite true, right? I mean, there are some academic scientists doing it. But then the question becomes, where are we in this? Artificial neural networks based on deep learning. I mean, before there was deep learning, right? Before we had like this humongous amount of data, and faster computers also were not successful. That's when symbol processing logic had its heyday in AI. So maybe today we're at a time when computers are still not fast enough. Maybe we need quantum computers or something to actually simulate the evolution of the growth process and neural networks. So not deep learning, but neuro-evo-devo learning, if you will, of artificial neural networks. And then they suddenly will become powerful. That's one thought. And just the last thought, and then you need to stop me because otherwise I keep on talking. It's also a big question of what we're trying to achieve. Arguably, to achieve a single task, I don't see why this enormous effort that I'm talking about here right now would be better. If you just need face recognition, it's, the deep learning is amazing. And I don't see how this like orders of magnitude more computational effort, evolution of a neural network would do that one task better. But this, of course, brings us to the question of, artificial general intelligence and where we're really going with this. Maybe you need that much more effort process if you want to go beyond single task artificial intelligence.
That was a lot that you just talked about. Maybe where we could start is the concept of unfolding information and algorithmic growth, right? So you had mentioned that there's not enough, well, you alluded to that there's not enough information contained in the DNA, in our DNA, to encode all the connections, synaptic connections in our brain. It's orders of magnitude less than you would need to encode the entire structure of our brain, but that through the process of genes becoming proteins and transcription factors, and through the developmental process, through which you call algorithmic growth, that information unfolds and that essentially encodes the program that results in our connected brain. So could you talk about the concept of unfolding information and algorithmic growth?
So the words are the best I could find. Unfolding information and algorithmic growth, but they're just putting a label on something that we clearly observe. We know that if we look at an apple seed, we can get all the information out that's in there. It's in the sequence of the DNA. Maybe some lipids around it are important. We need to know some physical laws. And based on experience that you've seen it before, that if you put that seed in the ground, there will be an apple tree one day. You know what it will look like. So in the apple seed, there is no way to read that it will be an apple tree. The only reason why we know that this is what happens is because we've seen it happen before. If you would not know, then I say the apple seed will not reveal this secret. You can only compare it to other sequences you have and what they did in the past, but that's the same sleight of hand, right? That's the same cheating based on previous outcomes.
Even if you could read all the DNA and know everything about the contents of the apple seed. Yeah.
Right. So this is what I'm saying, and this is controversial. Right. Clearly, there are more optimistic molecular geneticists than me that feel like one day, if we just know enough, we just will understand just how this happens. And so this is what brought me to try to find out whether there is any more solid way, any mathematical way. Is there something in science that tells us whether or not something like this can be unpredictable? That you have a simple code, right? Let's just call that 1 gigabyte of genetic information simple. I mean, it's not, but, it's amazing. But just for, in relation to describe, let's put it like this, describing the DNA sequence in our genome or in an apple seed compared to describing every single neural connection in your brain or even the branching pattern of an apple tree is like, it's like comparing almost nothing to an enormous amount of information, right? So clearly something happens. See, clearly during growth, there's more in your brain than you can read in a sperm and an egg. And so I tried to find examples. So I talked to mathematicians and I looked in other fields again. And so I came across something that has actually been, I've known for a long time, but it's not been, obvious to me where the connection lies. And I guess it's a connection that still requires some explaining. There are examples of very simple rule sets, very simple codes, that can lead to a lot of what we like to call complexity. And the example that I use then in the book are cellular automata, right? Mm-hmm. Stephen Wolfram made them quite famous with his book, A New Kind of Science. And he showed that there are types of one-dimensional cellular automata. I'm not going to go into details. There's super simple rules. You can do it on math paper. It's like, they're deterministic. They always produce the same thing. They're boring in many ways. But he could actually show that with a super simple rule set that I can write down in one line, if this, then this, if this, then this, if this, then this, done. And it's just black and white squares. If you keep on applying the same rule again and again and again, you'll grow a pattern that never repeats and that will literally grow with infinite time to infinite complexity. And for one of those rules, there is actually a mathematical proof by a coworker of Stephen Wolfram from the 90s already that shows that it is undecidable, which is math speak for unpredictable. So this is actually what is funny, not funnily, but I mean, this is what is called a universal Turing machine if it can contain in its pattern every single computation that you could possibly do in math. And this proof shows that. It shows that a very, very simple rule set can produce infinite complexity. It's the smallest known, the simplest known universal Turing machine in science today.
This is Rule 110, right?
This is Rule 110, the cellular automaton.
Which, by the way, I think would be a pretty good band name.
I guess so. Yeah, 110 is a number. That's the emergency number in Germany. Also,
oh, is that right? That would be a good German band name.
All right.
Yeah.
So,
sorry.
And it is, it produces infinite complexity, and we know that you can't predict what comes out of it. So what that means, I think I'm making it too complicated here. It's very simple. You can literally do this on paper, and everybody, in grade 1, you could already do this. Like draw line after line after line, and you will find that the pattern never repeats, and it's beautiful. And you may ask, well, could I have predicted? Is there any kind of math that would allow me, from just knowing the code, what the beautiful pattern in the end would look like? And now you see the analogy, right? This is what biologists and we all would like to know. Is there any math that would allow us from an apple seed or a human sperm and an egg to predict, without any previous knowledge of outcomes, what comes out of it? What brain wiring you would have? And so for this super simple mathematical concept, for this Rule 110 cellular automaton, we know there cannot be any way, any analytical math to calculate what, say, row number 1000 looks like. There's only one way to do it. You need to let it grow. You need to let an amount of information unfold that you then need, if you wanted to describe that endpoint bit by bit, would be quite a lot. But if you would only want to describe the information needed to grow it, would be very little. But you can't predict from the little information what the endpoint would look like without actually growing it. So this is why I talk about unfolding information. And this is why I call it algorithmic growth, which is, just a simple description of what we're seeing here, right? It's algorithmic because you use a rule set again and again, and you grow something, and there's no shortcut to that.
But so in the example of the cellular automata, this is a very simple system, right? And the idea in your book is that DNA doesn't encode the endpoint, but it encodes the algorithm to grow things. And DNA and the developmental process are way more complex than cellular automata. And one of the daunting things is that, let's say, we could go a lot of different directions here, but one of the daunting things is, let's say, your DNA encodes a protein. That protein has what we search for is the function of that protein, right? But through the time and energy in the algorithmic growth process, the quote-unquote function of that protein varies depending on different contexts and different stages of development. And of course, then you have all of these things interacting. So somehow the algorithm is encoded in the DNA and development takes care of the rest.
Yeah, isn't it beautiful? It's a problem. It's so supremely non-intuitive, right?
Right.
But we know it happens. I mean, there's no, there's no magic there. We know that you have a seed, you have an egg and sperm, and we know that given enough time and nurture, something beautiful will develop. And developmental biologists have just been studying how that happens, right? Instances of this, snapshots of this.
But in principle, one cannot look at the DNA code and infer the algorithm, right? So
we actually don't know that, right? So the reason why I like this simple rule 110, and you're right, it's of course much simpler. It's ridiculously much simpler to the extent that it really is not a good model for brain development at all. It's just an example that shows that a tiny amount of information, even deterministically, and the same rule applied again and again. I mean, the simplest possible thing, if you put in enough time and energy, can produce something of literally infinite complexity that contains everything, any possible computation. So my argument is kind of, if that simple thing already can produce infinite complexity, then we should definitely at least not be surprised that something that's so much more complicated, like a genome with a so much more complicated and prolonged and protracted developmental process, like, what happens for 9 months in the womb and then for many years thereafter, can lead to quite remarkable, what we like to call complexity in brain wiring. So what's so supremely non-intuitive about this is, where does the information come from? If the stuff in the genome is so little, and the information I need to describe the network connectivity is so much, where did it come from? And this is really the core of understanding the algorithmic growth process and the time and energy it takes. So there's a lot of beautiful discussions we could have now, and the physicists who are listening to this will know, of course, a lot about this, right? I mean, the fact that you can describe entropy in many different ways and find it, you can describe the information content of heat exchange, between my room and the outside and so forth. The time and energy you puts in information. So this is not easy to explain, but this is, of course, what we know happens, right? So it's not like I'm saying something outrageous. We know that, there's a seed, there's an apple tree. All you need in the meantime is time, energy, and, water and sunshine. So just information theoretically, what that means for brain wiring and how much information there is in a actual wetware biological neural network, I think should not be underappreciated. The amount of information that has grown into that thing while you were 9 months in the womb and while you were, growing up as a toddler and later teenager, with all the characteristics that you have at these developmental stages that we immediately recognize. Nobody mistakes a teenager for a toddler behaviorally. It should not be underappreciated. And neither should be learning. But, this is basically the trying to find the sources of information and then coming back to your original question or statement about pragmatic approaches in AI that, of course, must try to shortcut this, right? If you want to just make a deep learning neural network that recognizes, that helps your business grow, you want it to work. And so you're not going to go through all these processes, and you don't need to. But then the question is, can that thing ever be what our brains are? And that's where I think is no shortcut.
So you mentioned learning there and connectivity. So I'll ask about learning first. You consider, so the modern AI begins with learning, essentially. I mean, there's some, like you said, there's some dealings with architecture and how many units to use, etc. But then the almost sole focus in deep learning is the learning process. But do I have this right that you see learning as a continuum of the algorithmic growth process? Or do you see it as, is it separable? It's not like, so what I'm guessing is that you see our continued learning throughout life, and as we develop, these are not separable processes from the algorithmic growth process. It's all one big process. That's a question. Sorry.
Yeah, it's a wonderful question, and it's a big distinction between what we know about biological brains and artificial neural networks. Artificial neural networks, however close you want to make the design to the brain, they still have an on switch. And then there is this break you talk about, and then learning starts. So there's a design period, and then there's a learning period. Biological brains do not really have that. There is, of course, a period when you do not yet have learning. That's when the neurons are in a state where they're not yet excitable cells. They're not yet connected. Early in embryonic development, you have like all kinds of other developmental processes that have to, even start making that network. So you could say there is a break in the sense that there can't be learning yet while the connectivity develops. However, the moment the neurons start making connections, things start happening. Part of the developmental process of every neuron is that it becomes an excitable cell. They will start to spontaneously excite each other. We now know that a large part of very early brain development are activity waves that sweep through parts of a brain, both in an insect as well as in a human brain. And these activity waves are the brain learning from itself already, prior to even input. So part of the purely genetic program, no learning added, no environmental information added. Part of the purely genetic program is already that a neural network starts talking to itself, and that's part of what changes its connectivity even before you're born. And even before there's any input. The moment there is input, you're still inside the genetic program, in the sense that the way evolution selected for the genome that encodes the developmental program, is that there is a time when, for development to conclude properly, certain input is absolutely needed. I mean, there are horrific experiments I don't even want to tell you about to deprive a human of a certain input after birth. And then certain things will never develop. And there are critical periods, as they're called in biology, that if then the input doesn't happen as part of the algorithmic genetically encoded growth process at that right time, if you don't, if you're not talked to, if you don't get visual input, or if you don't get auditory or olfactory input, certain things can never even be made recovered. And they become part of the growth process. So the genetically encoded growth process continuously partakes and accompanies the learning process the moment the network is a connected entity. And this includes activity before there is any environmental input, before learning from any environmental outside information, before any nurture, if you will. But it also includes that nurture has to be right then and right there as environmental input, as part of that period of the growth process. And that continues our entire life in one way or the other.
It seems like such a delicate process that, there needs to be certain things happening at certain times within, or else the algorithm doesn't function properly, right? And yet, we also seem to be quite robust organisms. How do we reconcile those two things? Because, at first pass, doesn't it seem like, if anything goes wrong at the wrong time, in the wrong place, in the wrong environment, it could go haywire. And yet, we are surviving, thriving organisms.
Yeah, it can actually. You of course only see the winners walking the earth, right? All those experiments that evolution continuously makes that don't make it, just simply not there. The question of robustness is a beautiful question that neurobiologists, both developmental as well as functional neuroscientists, are struggling with. And there are certain features that we have learned that are key to the robustness of this whole program. And I think the most important feature is the idea of autonomous agents. The idea that an individual neuron actually knows nothing about the brain. An individual neuron has its own algorithmic growth process. It kind of, it grows an axon, like the big cable that needs to connect somewhere else. And, just when it needs a partner to make synaptic connections with, the partner happens to be exactly there, right? This is how the beautiful ballet of development unfolds. But if the partner were not there, the neuron would still run its program. And for example, in this particular example, by and large, a neuron would just make actually a synaptic contact with somebody else. So it would not be quite right, but it would probably be much better than nothing. And you can, early in embryonic development, you can do crazy experiments. So we work with flies in the lab, right? So in flies, when you develop part of the visual system, you can early in development easy go in and just kill half the cells. And then when you look at the final outcome, everything is perfectly fine again. So it just turns out that all the remaining cells during the growth process, they kind of just did what they normally do. But because there were no others that they would normally have had to compete with, which would have led to some of them dying and some of them surviving, they kind of all survive and they just fill the space. So this is very robust. And it's robust because it is not a blueprint-directed maker with some factory and robots that assembles it, but it is a self-assembly process of lots of individual autonomous entities. And each individual neuron, like every other cell in your body, has the capacity to encounter different environments, to encounter surprises when things go wrong during development, and when things happen, during normal function that is, of course, unpredictable in an unpredictable environment, and deal with it. And that's one of the key ideas that we know is important for robustness. And it's a very interesting concept because self-organization is therefore absolute key to the wetware neural network function of any brain, as it is for its development. But self-organization is kind of, kind of implicit in artificial neural network research, but is slightly avoided. So, you could argue that gradient descent and, parts of how backpropagation work and how neurons communicate has features of self-organization. And I think it does. But it's not a major topic in the design and training of neural networks when you look into the literature of the field.
I want to back up here. A lot of the book is dedicated, you mentioned the axon growth process. A lot of the book is dedicated to describing both the history and the current science and the controversies of how axons reach out and make the quote-unquote correct connections, although you were just mentioning that there isn't necessarily a correct connection from the start because these are autonomous agents, essentially, and they self-organize and end up and do okay. Before we talk about that process, thinking about the code of the DNA. So the book is all about the eventual connectivity of the brain, right? Brain wiring. But a lot of what is in the DNA must also be dedicated to metabolism, right? Because we have to survive in a far from thermodynamic equilibrium state. Do you see, is metabolism and the metabolic products, right, that are coexisting with the connectivity products in that algorithm, is the metabolic code separable from the connectivity code within the algorithm? And I'm asking you to speculate unless you have a definitive answer.
I dare say I have a definite answer. I'd say it's not separable. And it's not separable. This is a big discussion also in the larger now life community rather than AI community, right, about embodiment, right? The idea of, is a simulation enough? And how much do you need to simulate? Do I need to simulate the metabolic, things happening at individual synapses, or is it enough to just have, a value for a synaptic weight? This kind of thing. And if you talk about cells acting as autonomous agents, they start to have, of course, their own drives, and they need their own, at least minimally simulated metabolism. But this is at the heart of a self-organizing versus a designed entity. But you're asking about genes. And so if we look at genes, of course, people have been trying to find like individual genes that just tell us something about brain wiring, right? I mean, is there like a gene for this one synaptic connection? That's, of course, nonsense, because, we have, depending on how you want to count, let's just say 20,000 genes in humans and like some, ridiculous number of synapses in the brain. So how does this work? And so then people were trying to find, is there like, maybe it's like surface proteins that sit on one cell and on another, and then they recognize each other. And these proteins exist. But then you find that the same surface proteins, they're also functioning in the liver or, somewhere where like, blood vessels grow and need to branch and do things. And you try to find, okay, this metabolic stuff, is this just, is metabolism only like kidney, heart, and liver, or how about the brain? And then you find like, yeah, of course, most metabolic enzymes are actually expressed as particularly high levels in the brain. And it's the organ that requires and uses most energy. And so basically the bottom line is that, there are a few heart-specific genes and there are a few brain-specific genes. But by and large, this whole question is not very good. The idea of hoping to find a gene that specifically tells me how the brain wires. There are very few genes in the human genome that will not be turned on and off, that will not be read out in one way or another, in one cell or the other during brain development. And it's all part of the algorithmic growth process. Evolution didn't care about our intuition of, say, two molecules that recognize each other and could be a key and lock for synaptic connectivity. Even though, we love to write papers like this as developmental neuroscientists. Look, I found another cell surface protein that's exactly on that cell, and the lock to that is another protein on the other cell, and, both are genes in the genome, and this is how this one synaptic connectivity is wired. But evolution, I dare say, really tried out any kind of mutation in regulatory sequences and coding sequences of any kind of gene. And as the growth process unfolds, you will find that a mutation in some ubiquitous, that's, biology speak for expressed everywhere, expressed metabolic enzyme, a mutation in that thing that, let's say, changes just, it increases 5% of the function. That'll turn out to be completely irrelevant for your heart and your kidney where that enzyme is as well. But during brain development, there may be just this one neuron that if you increase this particular metabolic enzyme 5%, at the exact time when it is making a synaptic connection, it changes the speed, say, because of the increased metabolic rate, of the time window. So it shortens, say, the time window of when it can make a certain connection, and it will lead to less connection of one type and more connections of another. And that was a mild change to a metabolic enzyme that's everywhere in your body. And the only change it may cause in the outcome, completely unpredictable, but, evolution tries these things out, was a slightly different wired brain. So this is why you need all of it. You need, you can't just take a synaptic weight as a number, but the information encoded ultimately in all these synaptic connections and the way to get there required an evolutionary process, selection of something that worked, that was not predictable, like Rule 110, but that, if you had enough millions of years to try out evolution on Earth, and evolution of brains, you can figure out adaptations and changes based on mutations in many different genes. And there need be nothing intuitive about it for what scientists would like to see.
So modern deep learning, right? It begins with a network of connected artificial units, and like we've been talking about through a long process, the synaptic weights get adjusted through learning. But it starts as a neural network, and a large part of what you describe in the book is the developmental process that leads to a network, which is a bunch of neurons connected. And you give multiple examples throughout the book of different connection patterns that can happen depending, on the sequence of the development that happens. And yet, the focus is still on sort of the end connectivity of the brain, that you end up with a network of neurons with connections between them. Are neurons the end all, though? Do we need to consider things beyond neurons, like astrocytes and glia? Is that part of the whole algorithmic growth process that may eventually be important for building better AI and understanding how this all works? Or do we really just need to focus on the network of neurons?
So again, we need to come back to what we want. What is it that we're trying to achieve? To make an algorithm to predict what to buy, I don't see why you would need astrocytes. If you want to have any kind of resemblance to what you may want to call a human AI, I'm hesitating because I'm trying to use the word general artificial. There's so many terms. Yes, they're all undefined, right? I mean, artificial general intelligence, nobody knows what the hell that's supposed to be.
Yeah.
And that has a lot to do with there being no definition for intelligence. And, the idea then that you want to have a human intelligence is very different to me from human level. I don't even know what level is supposed to mean, right? I can measure a level on playing chess, but I cannot measure level on being, Paul. How Paulish is my intelligence? And I think many different people will be more similar or less similar to your individual type of intelligence. And that requires your entire brain.
One of the seemingly important features of brains is the feedback loops that occur within if we can just stick with neural networks. The brain is a highly, highly, highly recurrent neural network, and AI is taking this on. Deep learning is taking this on, and people are using recurrence. But my question is, thinking back to the algorithmic growth process and a self-organizing system, and actually in the book you talk a lot about levels. So going to the DNA level, then it is interesting, and I'm going to ask you if there's a deeper principle involved here. It's interesting that an enormous amount of DNA is devoted to feedback in the form of regulatory proteins that feed back onto the DNA and regulate what's being encoded by the DNA. Transcribed, I should say, harking back to my own molecular biology days, trying to remember the words. And then through the developmental process, it seems to be that these feedback mechanisms are also, like a majority of the processes. So is this a deeper principle that within algorithmic growth, to have a robust system, that feedback is the main thing?
I think so. It is a very important thing. I mean, this has been recognized. You can go back to the old cybernetics days, right? Mm-hmm. This is Norbert Wiener and others who first formulated and quantitatively worked on ideas of feedback and how they determine self-organizing systems. Very specifically to the example you give, it going back to everybody's biology 101, right? The genome doesn't change by and large from once the sperm has met the egg, you're kind of done. I mean, that's your genome, and every single cell in your body has it, and it's the same genome in every single one of your cells. And of course, what makes a cell in your eye different from one in your heart is that different parts of that genome have been read out during a developmental process. And that process, as we already discussed a little bit earlier, never stops. We know in the brain, when it comes to learning, that to form long-term memory requires the feedback going back all the way to the genome, new transcription of the mediator of what will become proteins, the famous RNA, and then making new proteins. And then those proteins get incorporated into whatever molecular function you need to change the physiology of the cell. Many of the proteins that get this way read out from the genome are proteins that themselves have this funky property of binding back to the genome, which then leads to yet another different type of protein being expressed. And, maybe it's not one, maybe it's a different 1,000 again. And then one of those 1,000 is again one that binds back to the DNA and changes it. So both the internal program that keeps on running changes the cell continuously in a feedback process, and that's part and parcel of any developmental growth process. But also, the environmental input, once you have a neural network, will feed back to that genome. And the genome is always just there. It's like a book that's always there, and you always just need to decide what to read in that book. And to access that book is just enormously complicated. You can't just open page 255. You literally need a very strange combination of, say, 30 different proteins that are super unlikely to ever exist at the same time in the cell. But if they do, then a particular gene combination, these 231 genes or something, will be transcribed, and you will have a new state of the cell. And this is, of course, what happens all the time. And none of the cells, very few cells. There are a few cells that actually are very silent in our bodies. But most cells in our bodies never, ever stop that feedback process, right? If you have an injury in your skin, it goes all the way back to changing what will be transcribed. Cells in your heart, and of course, cells in your brain. So the idea of a feedback to the genetic information and what will unfold as a next step. This is basically what all of biology and all of biomedical research is about. We're continuously studying when and how, what kind of genes get expressed. And this is such an enormous field, even with this only laughable 1 gigabyte of base pairs in the genome. That's not much information if you want to write it down. Easy, 1 gigabyte. But the information that can, just in 1 or 2 iterations of any of the 20,000 genes that are being expressed, change what will be expressed next, is just a combinatorial explosion that gets out of hand, like almost immediately, and kind of ensures that researchers will never be out of having something new to study.
It makes sense that AI researchers would want to avoid all of that mess.
Yes. Which is why you can't design it. So the only way you can deal with that mess is you have to give up control over the design. You have to program it, literally, by making random mutations and hope for the best. And if they're not good, then what comes out of it will be just not as good in the outcome. And evolution didn't know, but evolution will just select against it. And if what comes out of it is better, then, you keep some of these new randomly tried out mutations and you program something better. Because, remember Rule 110, if, that's a proposal, but, we're just take it for what it's worth now as a hint that it may just be just as unpredictable how the genome encodes, what comes out of a given genome without having prior outcome information. There's no other way to program this. And, if you do this, then it would be nice to at least keep it, not hyper multidimensional, right? To at least have only a few genes and a few interactions. And, then it's still a lot of computational effort to simulate that. To basically do this experiment and for every single slightest random change to see what comes out of it, to do this entire effort of, 9 months in the womb and then, all those crazy teenage years where you just don't know what you want. And then finally sitting here in a podcast, that's not what you would like to do if you have a pragmatic job to do in programming a neural network.
In the book, I guess you get a little philosophical about generality and specificity, talking about the growth and development process and how different proteins are used in different contexts at different times in different environments. And what we want to do as humans to understand what's happening is we want to have a very general principle, right? But then it's really difficult to say, well, you know what? I'll just read what you write in the book. This is a bit of a conundrum. We can choose between fewer general mechanisms that explain more instances less, or many less general mechanisms that explain fewer instances better. Where we draw the line for generality is anybody's guess. And this is a recurring theme of, you also talk about levels and what's the right level to explain a given system and so on. I don't know, could you just comment on that balance between generality and specificity and that conundrum?
So in our field of developmental neurobiology, it's funny how papers are written, right? I mean, of course, everybody is studying like a super specific system. I mean, we study flies and some other people study mouse and a specific neuron at a specific developmental stage where it makes a certain choice and you understand how molecularly it does that. And so, of course, every single one of these scientific publications about a process like this then has to say, we are looking at this super specific thing. But then the hope is, of course, always, we're looking at the super specific thing, but really, it's very general, right? I think we found something in our specific instance of the problem that tells us something how this generally works. And so, everybody does that. Everybody then writes like, this gives rise to a potentially general principle of brain wiring. And the classic ones are, of course, the attractive or repulsive molecular interactions. And they're clearly part of how neurons interact and how brains are wired. These are words that scientists use, but of course, generality is, as the sentence you just read, I hope, kind of suggests, they're not black and white. I mean, what does it mean? Like, this is a super general principle. I mean, I can give you the super general principle that everything that happens is read from DNA into an RNA and then into a protein, and then the proteins interact and zoom, you got a brain. That's a really general principle. And everybody will agree. I mean, this is what happens. The feedback stuff that we've been talking about. This is a general principle. It's a general principle that you have continuous feedback of the proteins, which are the products of the genome, onto the genome itself to change what next will be read out from the genome. General principle. But of course, if I phrase it as general as that, it tells me very little about how this one neuron made a synaptic contact with another neuron. So there, I need to become a bit more specific. And then the question is, how specific do I have to be about every single molecule that is there right then and, at the right place and at the right time to put this thing together to understand this instance of the problem. And then still I want to say, but this is now a general principle. So it's just I'm not sure it's very philosophical. It's just an observation that we have when we're trying to understand any system, right? And you can always make a very general statement that's almost certainly true, but really then not very helpful anymore. Or you can make a very, very specific statement that's really helpful to understand that specific thing. But then, of course, it's not going to apply anymore to every other, So there are things that every neuron has, every development of a synaptic connection has in common. And then there are things that must be different. In the end, the idea that you need all of it, that you do need every single one of those molecules that have to interact, that unfold specifically at some point differently at every single synapse in the brain, such that every single one is in some way different from any other, is irreducible if you want to have that thing in the end, the brain. And so the selling point of, but we're looking only at generalities, is helpful to a certain extent. But at some point, we just have to appreciate that it's really an arbitrary choice how deep we look at any given synapse and at any given neuron.
Well, toward the end of the book, you argue that, so you talk about whole brain emulation, and you also talk about the brain AI interface that is oncoming with current companies trying to do this, like Neuralink, etc. But you argue that to emulate a whole brain, you actually need to do it start from the molecular level. You need all of those details, right? How strongly do you believe this? And have you received a ton of backlash on this?
How strongly do I believe? I mean, the data just shows. I mean, this is. I try to avoid to believe. I try to believe anything really, but I'm very happy to be proven wrong. The biological systems that we study are just that, like that, right? If you take out any component at any point, you have surprising implications. And as we discussed earlier, this is exactly how evolution programs a brain. So if you take any component out, if you simplify it in any way, you can still get something that's amazing, but it's not going to be that thing anymore. So if you want human intelligence, the argument is you need every single one of those molecules. You can't take any of that away. That doesn't mean that you can't produce something more intelligent than a human in many other ways. It's just not going to be a human intelligence. So, in terms of backlash, the idea is, of course, not very well received by the more pragmatic camp of deep neural network developers. But they don't mind really all that much because, they know that what they're doing is amazing. They know that what they're doing is successful in what they're trying to do. And, it becomes a little bit philosophical again then to talk about where the whole journey is going. I'm not actually clashing with any of these amazing people who are developing neural networks. I think we're all equally impressed with those. Where I am genuinely clashing is with the prediction of where, therefore, inevitably current technology of neural networks has to go. The notion that clearly the next thing that happens is an even more intelligent thing, and the more intelligent thing will have this enigmatic property of being able to produce a yet more intelligent entity, and therefore then we have superintelligence and runaway intelligence, and we have the famous omega point, and we have singularity, and then they're all going to take over. So, I disagree. We're not just far from this, we're just not even anywhere near the right path to anything like that. And the argument has a lot to do with what we understand about what it means to make an intelligent system. The key argument I would make against this whole singularity debate, if this is where our journey is going right now, on which I'm not an expert at all, right? But I mean, I'm coming from another side, and so I'm just raising a voice, a critical voice here, and is that there's no example anywhere in the known universe of an intelligent system producing a system that is more intelligent than it. Our entire discussion that we just had circled around the idea that evolution is the only thing that can program a thing like our brain, precisely because of the unpredictability of Rule 110 and what the outcome is, and therefore you need the entire unfolding thing. So the notion that just because something is even more intelligent than us, it will automatically, inevitably have that enigmatic ability to produce a yet more intelligent thing. I don't see why at all. I mean, why are we not able to make something more intelligent than us then? And we're not. I mean, there is no example of that in history. That's the first major criticism. And the second major criticism, if you read a book like Superintelligence,
I don't know that I recommend reading that book. It was a rough one for me.
Interesting. I agree with you. I mean, but it has some. It tickles the senses and the excitement about the future, as it has always done, right? I mean, there is entirely, absolutely unproductive branches of so-called science that have been dealing with this ever since Werner Winge and this whole, superintelligence thing came up many decades ago. They haven't produced anything. They've just been talking a lot and making a lot of money. But one of the things that, for example, Bostrom desperately needs and just glances over in like 3 sentences in order to copy your brain is just a brain scanner, right? You just scan your brain and you get the information out, and then you can take that information and make another one. Well, we have just been discussing, you and me, the entire time that we don't even know what information we need to what molecular level in order to have a brain that actually functions. So what is it that you're supposedly scanning? In theory, at least, which is practically absolute pipe dream, but in theory, at least, you could do like a molecular scanner that takes every single molecule and makes a copy of that, and then you have that other brain. But that's of course not what they mean. They all mean that, yeah, clearly it got to be enough to have something like, say, states of synapses and some digital representation thereof. That's a pipe dream.
So I'm taking it you're not a fan of the idea that one could slowly, let's go with neurons, right? Replace our neurons with mechanical neurons very slowly, and then we would have a functioning brain that is half, or however much neurons make up our brain, right? That wouldn't be functioning well. That we're not anywhere near the sophisticated level that it would take to create an artificial neuron that would be able to function holistically. Well,
I don't want to be as critical as that. I'm actually very optimistic and very impressed with current machine-brain interfaces. Mm-hmm. As you know, we can now do extracellular, little electrodes and thousands of them into, say, somatosensory cortex. And that'll allow somebody who has lost the ability to move an arm or a leg to move a prosthetic device. And this kind of technology is super. I mean, it's fantastic, and it's going to improve, and it's going to get better, and it makes perfect sense for companies like, there's the BrainGate initiative, there is Neuralink, that, of course, because it's Elon Musk, made more headlines, but it's not really all that new even.
No, it's not. You didn't roll your eyes there. I was expecting an eye roll. I often
no. I mean, I'm actually trying to be positive here, right? Yeah. I love these approaches. I think it's awesome that they're doing this, and of course, what it's designed for, right, is if we just leave the science fiction out, is at the moment tetraplegia, like patients who really can't move their arms or legs, and the locked-in syndrome patients and so forth. If you can have any bionic device that communicates with the brain, that helps these patients, bring it on.
Yeah.
And if you're blind and you can put a little camera in front of your eye and connect it to the visual cortex, and you can, you're not going to see as you and I are, but you see at least something again, it's fantastic. And there's no reason to not assume that this is going to become exponentially better in the next years. I'm sure it will. It's going to be wonderful. And it's going to come with its own challenges, and there's going to be some critiques, and, are the Borgs now finally coming? And there's going to be some concerns, and there's going to be some failures, and there's going to be some successes, and how any technological advance goes, and it's all good. It's all part of it. So this is going to happen, and it's going to be great. But this is not what we had been discussing, and this is not what Bostrom talks about when he has a brain scanner to make a copy of your brain. We're talking about, this is, do you even know what kind of information you would want to get out of a brain to make that brain, or to make any brain? What makes Paul Paul? Can you just copy that and then another person can be Paul? This is a whole other level. The amount of information we're talking about here is something that we really just don't even understand.
Yeah.
And if you just want to bring it on a very technical basis, right? I mean, when we're talking about a few thousand electrodes and all these things too, it's super impressive. But, these are extracellular electrodes somewhere in a brain region where no one knows what the hell these neurons are doing. Individually, where individual neurons can die and everything is fine, and you just get field potentials, as it's called, from like these areas of the brain. This is not the same thing as neurons that are intricately wired via all the molecular glory of synaptic connectivity inside an actual brain. They're valiant approaches and efforts, and they're to be applauded, and they're going to be big successes, but they're not a path to making a human brain.
So, Robin, there are a ton of other topics that we could go into from the book. I'm aware of the time, though. One of the cases that you make in the book is a case for the utility of cognitive bias, right? So I'm not going to ask you about that, but what I'm going to ask you about thinking about cognitive bias is during the process of writing the book and thinking about these things. And, in the beginning, we were talking about what led you down this path. Has your view of intelligence changed through this process, or is your cognitive bias stuck where you had the same view of what intelligence is and what it means, et cetera, before you were writing the book?
The embarrassingly safest answer is that I can't possibly know, because I'm sure I'm somewhere stuck, right? And I'm, caught in my own brain, in my own biases, in my own growth history, right? So I come to the topic of biases through the growth history, right? Which is serendipitous and idiosyncratically individual, and makes you and me. And of course, new information coming into a brain is not compared on an even footing with the information that is already there. I mean, we know this. And that has a lot to do with how you have used your brain in the past. And let's just not go into all the examples that leap to mind right now about data unbiased beliefs that people have right now. This is how the brain works. So, this is a dangerous area to be in. They actually, this is not my field, but there are amazing experiments in psychology out there that actually show that people who are trying to not think about these things and people who are not trying to worry too much about themselves are actually more successful. To be ignorant to your own, you can look up these things like the embarrassing questionnaires. So it's a very difficult and dangerous area to go in, right? You can go and spiral down in self-doubt very easily if you question data-based every single part of your growth history based on a brain that happens to function based on that growth history. So has my view of intelligence or any other aspect in the book changed?
Yeah. Well, the way that I asked the question, it sounds like I'm asking, what was your view before and what is your view now? That's not actually what I'm asking because, like you've already said, we don't really know what intelligence is. What I'm more asking is, do you feel like your approach to understanding intelligence has changed throughout the process? So you can answer, I thought this before and now I think this, but that might be an impossible question, of course.
I think I've become more humble. I mean, if I dig into my idiosyncratic past, I remember being a young 20-something-year-old scientist who thought he knows everything.
But that's what got you to where you are.
I've never been more insecure than ever in my life than I'm now. Maybe that's a good thing. yeah, it got me to where I am. Exactly. I'm humbled. The process of writing that book and making the connections that I try to make is a humbling experience, because you have these moments when you realize parallels in history. You see that all these things have happened before, and you find these references in people who have thought these thoughts before. There's so much out there. There's so much beauty and so much knowledge in the literature. People who have, if you just go into what people thought, I could have gotten completely lost just in the cybernetics years.
Yep.
Between the 40s to 60s, and all the thoughts that people back then had, like Ross Ashby and so many others. And it's humbling, right? Because then here you are, and you're writing this book based on, there got to be something that we need to talk about, because we should learn from each other in the AI and neuroscience communities, based on the idea that, we have a genome and you don't. And then you dig into this, and it's of course a bottomless pit, and so many thoughts have been thought before. And I'm, with every minute that I'm spending on a project like this, changing my brain in a certain direction and not another. And time is so scarce.
Yeah.
I'm running out of time every day, as I guess we are right now in our conversation. If I want to investigate something, I need to write this chapter. The temptation to just get lost and dive into this almost infinite depth of knowledge that is out there is very humbling. And so then you kind of, I find myself then disciplining myself and saying, okay, I have 1 week for this thing. And I have 1 week to interview people and to learn about this. And, this is how far I go. And of course, if you do it well, you must be, after this 1 week, at a state where you realize, oh my God, there's so much more
Right.
than I initially even thought that could be known, that it must necessarily be humbling.
So one of the things that you did allow yourself to dive into in the book is Roger Sperry and his work, and also Mike Gay's, and you talk about their differing ideas in the book and go into some depth about it. Maybe the last thing that I'll ask you, you talk about how there are differences in scientists, right? Some are more vocal and some are quieter. Mike Gay's was a quieter scientist who didn't self-promote as much. And not necessarily that Roger Sperry self-promoted, but he was more opinionated and willing to vocalize those opinions more. Both great scientists, right?
I think it's fair to say, yeah.
Yeah. The question is, how self-promotional as a scientist should one be? So now, I'm asking someone who's written a book and needs to promote it as well, right? Or, hopefully we'll sell some copies, right? But is it better to be wrong and popular or well-known? Or is it better to be right but unknown? Some career advice for those.
Oh my God, this is so difficult. I can tell you from the bottom of my heart that I did not write this book with the intent to, make it a New York Times bestseller. I would like it to become, and I'm actually delighted to see that it is being picked up more than I initially could have even hoped. So it's been doing very well, and I'm very, very happy about that. But, writing the book is one way of being vocal. But the example of scientists you give right now is also about just how you present data. There is no way in science to present data without interpretation. Mm-hmm. And the way you present it, the claims you make, bringing us back to the discussion we had before about generality, but also the manner in which you do this has an enormous influence on how it is perceived. And I'm afraid that there are many examples in the history of science where a really good idea and a person who actually got something right has not been corrected to this day. We always say that science self-corrects in the end, and I believe, to the extent that I'm capable of believing anything, that is likely true in the limit of infinite time. But it's scary how many processes, many ideas, right, have not been corrected, and people actually got forgotten, although they said the right thing,
Right.
For that reason. So this is very tricky. I can tell you that I basically did everything wrong. I did write the book, so I maybe that's, in itself something right, but because I always felt that science should be something that is promoted by the value of the science itself, which happens through scientific publications and peer review, and then reviews about that work and discussions within the scientific community based on scientific publications themselves. I, until I think 2 days ago or so, didn't even have a Twitter account.
Oh, you're on Twitter now.
I'm not really. So we made an account for my lab. So we just published a beautiful paper, 21st of December. And so I decided, the Hiesinger lab has now a Twitter account so that we can at least tell our colleagues we published this paper. But This is literally, I've sent one tweet in my entire life. That's a really bad idea in this day and age. If you want to write a book and promote it, and if you want it to sell well, you better have a good following. You better have a Facebook presence. You better have a LinkedIn account. You better have an Instagram following. And I literally have none of these things.
That's great.
Not even LinkedIn.
You've caved in now.
Well, so we see, this is the question. Yeah, how morally, I don't want to be morally superior, right? I mean, I don't want to, it's just choices we make. And so, there are ways how you can try to sell whatever you want to sell more. And clearly, I have not tried. I mean, there's no advertisement. There is no there's no tweet. There's no advertisement. There is no Facebook. There is no Instagram. There's nothing about this book. Other than that, here it is. This is what I wanted to tell you. I did this bit of work. I put all my heart and a lot of work in it over 3 years, researching the history and the current state of how developmental neurobiology and artificial intelligence see the information problem. The question of how does information get into a neural network? How does a neural network grow smart? And this is what I wanted to do. And I wanted to contribute this, and I'm proud of it. And now, live with it. And if somebody wants to read it and it develops its own life, you contacted me and you're asking me questions that belie, a deep knowledge even of many of the ideas in this book. And so that makes me super happy. And maybe that's better than a short tweet,
Well, Robin, you should be proud of the book. I didn't mention the way it's laid out is a series of 10 seminars. And before each seminar, you have these different characters like neuroscientists and a robotics engineer and various personalities dialogue with each other. So in Gödel, Escher, Bach, Hofstadter uses this kind of form, but it really doesn't work. Like it was with the tortoise and the hare, I believe.
Oh, really?
Not for me.
I thought it.
You liked it?
Oh, interesting.
To me,
I liked it actually. Yeah.
Dragged
but
on.
honestly, I have to tell you, it's been 20 years ago. It's been 20 years since I read it, so
Okay.
Maybe I should look at it again. It's a bit different. So what Hofstadter did in Gödel, Escher, Bach is that he does this really fancy, funky thing where they actually talk like like a canon of Bach or so, right? So it's very symmetric, and it kind of it also just probably serves a little like relaxation, right? Purpose that between like heavily loaded chapters, there it's nice to relax a bit. In the self-assembling brain, the dialogues are different. They have a different origin and a different idea. The reason why they're in there, they were not in my original book proposal, and they were not in my original draft. The reason why they're in there is because this is what my notes looked like.
Yeah.
So when I got dissatisfied with my own field as we started this discussion, right, and I felt like it can't be that we're just studying molecule by molecule, building a parts list, and we don't even know what the endpoint is. And I'm not sure what we're missing there, but I wanted to know what could be out there. So I just started to go to other conferences. I just, went to an artificial life conference and just, let's see what these people are saying. right? And it was eye-opening. It was beautiful. And I met amazing colleagues that I'm working together with right now. And I had discussions with them. And so then and of course, then when you already have the idea of the book in your mind and you go to this conference and you just, I chatted up people. So I targetedly I go to you and then I just, start to have a discussion. And of course, I ask them certain questions and then, some people just are amazing and you just get into this. And then I would always run back to my hotel room and just write down the conversation. And, so as to not to lose the arguments, as to not to lose, I asked him this and then he said that and I asked her that. And then she actually said, that's not even a good question. You need to do this, this, and that. And so it was very clear that, people are always talking based on their own growth history in their own worlds, mm-hmm. right? I mean, in the beginning, these discussions are very tricky, right? Because I'm asking one thing, but I'm getting a completely different answer in vocabulary that I'm not necessarily understanding. And then I did this in my own field and, I could ask the same questions. And so I basically started to I had a lot of notes from discussions and all these characters were there. There's the robotics engineer, there's the developmental geneticist, there's the neuroscientist.
Was that more fun to write, easier to write, harder to write than the rest of the text?
So this is where I'm just getting it, right? So the initially I didn't write it like this. I tried to get the essence from my notes and I tried to write like a normal book.
Yeah.
And it was very difficult because what you lose is the way they talk coming from a different place, mm-hmm. right? If I say something as a developmental neurobiologist and try to describe something and a deep learning expert tries to talk about it, we talk differently. And the way we talk is just loaded with our own history and our own way of talking. And the way we even then try to communicate becomes quite interesting. And it takes time for us to find each other. And so this is what happened, right? So I had these notes, I had a discussion, I tried to distill it, but, just to circumscribe basically where people are coming from is so much harder and cumbersome, and it didn't read well. That in the end, I just felt like, just let these people speak. And so I just started to write the discussions between those people, and it was much easier. Because then, of course, the developmental geneticist can say these outrageous things that I wouldn't even as an author dare to write in a book because, I think this is like, hardcore. It's too much. It's not that deterministic and so forth. But as a character, I can have a developmental geneticist say all these crazy things because that's what people are saying. Yeah. And then I can have a robotics engineer, of course, come in and say, you know what, we don't need all your wetware and that's all nonsense. And we can do this all much better. I don't need a bird with all this stuff. I can make an airplane that flies much faster. But to say these things is best said if you have characters who are themselves and have their own growth history, if you will. And then, of course, that became a beautiful challenge over 10 dialogues to have them find each other, right? So in the first dialogue between them, they're all talking cross purposes, right? They're all just, nobody understands each other and they don't even appreciate each other. And so, of course, what I hope happened to them is that in the end, they understood each other a bit better.
Well, Robin, thanks for writing the book. Thanks for unfolding information with me here today and spending your time and sharing it with the podcast listeners. You'll be getting a tweet from me soon about the book.
And you know what I'm going to do? I'm going to retweet that from my account. That might just be the second tweet that I will ever have sent him.
You are on top of your game, sir. So thanks, Robin. It's been fun.
It's been awesome. Thank you very, very much.
Brain Inspired is a production of me and you. I don't do advertisements. You can support the show through Patreon for a trifling amount and get access to the full versions of all the episodes, plus bonus episodes that focus more on the cultural side but still have science. Go to braininspired.co and find the red Patreon button there. To get in touch with me, email paul@braininspired.co. The music you hear is by The New Year. Find them at thenewyear.net. Thank you for your support. See you next time.
The